Welcome to our YouTube series. My name is Ramji Srinivasan, co-founder and CEO of Teiko. We are going to talk today to you about fluorescence minus multiple, also known as FMM, and explain some of the basics of this very powerful technique, how it works for validation in cytometry assays. And just as a reminder, our mission at Teiko is to make high-parameter cytometry for clinical trials simple, easy, and routine. You can download some of our validation reports that include FMM at teiko.bio/pbmc-spectral-flow as an example. So, who cares about this subject? In short, any drug developer who wants to ensure the cytometry panel they're using is precisely detecting what it is intended to measure. And that seems like a tall order, so let's be a little bit more specific. So in particular, let's look at the APC fluorophore on a spectral flow cytometer. So here you're going to see a chart of an APC fluorophore across several emission detectors. On the x-axis are the different colors of the spectrum, everything from ultraviolet to red. And over here are the individual channels, UV10 through UV15, YG3 to YG10 in yellow-green, R1 through R8 in red. And on the y-axis is the relative emission. And what you're seeing here are the conceptual drawings, but the actual machine records a digit showing the signal intensity for that underlying detector. For example, for APC, over here it might be 1, for UV10, UV11 might be 5, and over here where there's higher signal, this could potentially be 40, 25, 1, and so on and so forth. So the issue happens when you're comparing multiple fluorophores, the emission spectrum will overlap. So we just added in Alexa Fluor 647, and you can see that emission spectra is also in this yellow, green, and red area. And as a result, what you need to do is validate that the signal from these markers doesn't what's known as spill over onto other channels. So how can you measure the level of spillover? The short version is just take that marker out and make sure you don't see signal in that channel. So in an ideal panel, if you remove this marker, you shouldn't detect it. So imagine you run a sample with this removed marker for APC. As you look across, you shouldn't be able to see much in this particular row. In fact, that's what we're seeing. So take the marker out and make sure you don't see signal in that channel. So in an ideal panel, the remove marker shouldn't be detected. Let's take APC and assume that it's been removed. So you can see on the bottom over here, you don't see much signal in APC as you go across all the detectors. It's basically 0 or pretty much close to it. But if you add another fluorophore, this is when you might see spillover. For example, if we added Alexa Fluor 647, it's in the yellow, green, and red areas. Over here, we see that APC had been bumped up a little bit. This is what's called spillover. Basically, what's happening is AF647 might spill over into the APC channel, adding to that signal's intensity. We're going to zoom in on the yellow, green, and red parts of the spectrum, so that's a little bit more clear. Let's zoom in on that bottom right part of the spectrum. For example, the yellow, green, and red parts, before Alexa Fluor was added, it was in the 0, 1, 0, 0, 0, 0.5, 0.20, and then after, you can see a bump up in the signal intensities across these sets of detectors. So the spillover is the magnitude of this additive effect. And to go in that more detail, we are going to show our 25 marker spectral flow. This is the panel we ourselves use at Teiko. Obviously, you can remix and feel free to use your own. And normally, to quantify this additive effect, you would begin by removing this marker and measuring its signals. For example, getting rid of CD127. And this is what's called fluorescence minus one, or FMO testing, which is well known in the field. Although it would be great to do this one by one and go through every single marker and then just cross them out and see how much signal it is, it's very expensive and laborious to complete this. In fact, as you go from a 4-marker panel to 25-marker panel, it's about a 6-fold increase in cost and potentially time as well. So as a result, this is where fluorescence minus multiple, also known as FMM, has emerged as a technique, first described by Wannick et al. In 2021. In this case, you would have a normal blood sample, which would have a 25-marker panel. FMM1 would be the 25-marker panel minus 6 markers. FMM2 might be another 25-marker panel minus a separate 5 markers that are not in the 6. FMM3 would be a 25-marker panel minus another 5 markers that are not in the 6 or 5, and so on to FMM4, which is again the 25-marker panel minus 5 markers that are not captured in any of these. So to make that clear, let's go through the original panel again. So we have this 25 marker panel over here. We have FMM1, and you can see all of the missing channels over here. So these are the 6 markers that have been taken out. FMM2, a bunch of other markers have been taken out, but you can notice none of the same ones are taken out. FMM3, and again, we've taken out 5 markers here, but they're not the same ones as FM1 or FMM2. And then finally, FMM4. So same logic applies, which is the markers that are removed aren't the ones that show up in 1, 2, or 3. So the name of the game here is, for example, with CD141, you really want to make sure it doesn't show up in your FMM1, otherwise something has gone wrong. Or HLA-DR, you want to make sure that doesn't show up in FMM2. So some design considerations across FMM controls: you want to distribute nearby fluorophores, you want to distribute activation markers. What you want to do is place marker pairs used in bivariate plots on different FMM controls. We'll go through a specific example. Exclude primary lineage markers that show consistently clean separation from the controls, and generate the control by combining all antibody conjugated fluorochromes except that of the absentee markers for each FMM into a single cocktail. And this is all adapted from Wannick et al. Cytometry A 2021. So let's go to that bivariate plot example. This is an example of gating for CD4 positive non-Tregs, and the two markers that are relevant here are CD27 and CD45RA. In a bivariate plot, you would see these, say, CD45RA and CD27 like this. And what we're going to do is put these on different FMM controls. So you can see here for FMM2, CD45RA is excluded, but it's included on FMM3. And then conversely, CD27 is excluded on FMM3, but included on FMM2. So this goes to the underlying result. So we have a 25 marker panel. We then looked at the marker positive in the full panel. And then for our own internal results, we looked at the marker positive in the corresponding fluorescence minus multiple. What you're aiming for is low values in all of these. And lower values are better because you don't want to see a signal in a place where there isn't a marker or it's been removed. The threshold we set was 1%, but obviously you have to set the threshold that makes sense for your experiment. Plotted on an XY plane, so CD45RA to PD1, and the y-axis is percent of total live cells. And then here's a 1% acceptance criteria. So you see all of these are well below that threshold, which is great. Remember, the lower numbers are better. And so just as a reminder, the goal of this is to ensure a cytometry panel is precisely detecting what it is intended to measure. FMM is really well designed to solve this problem, and hopefully you heard some useful design considerations. So if you're interested in more detail on validation in cytometry, subscribe to our YouTube channel to learn more. Thanks for stopping by.